So what we're going to talk today about is we're working our way through the last section, which is 3 booklets. So it's a quite amount of stuff. And so what I did for that is I made a very extensive test review for the physiology type stuff. And for the anatomy type stuff, we have our test term list number 3 in the anatomy labeling. You've been starting, I've seen a lot of you guys have been doing the heart already and are working on the blood as well and keep moving forward. So that's perfect. So we want to go a little bit through what I have on the test review, and then we're gonna do a little bit of a labeling quiz, just a few terms on the heart, so we get that process started. Again, that's going to be like last part as well. Hi, Zara. And I have Jamuna also. Hi. All right, let's just dive right into it. No, that's on the PowerPoint, I believe. Hello. There we go. Can you see my PowerPoint?
Yes.
Yes.
Good. I have to redo some PowerPoints. All right. When I look at the, and then of course the test review, I should show you that first. Did everybody, were you guys able to print it out?
I haven't yet, but I'm sure I could.
So, or at least have it somewhere downloaded, or it's one of those that I think is a good document to print out because you can take notes on it. You can refer. When you take the test, it's hard to, on the electronic devices, for me at least, to jump around on, I like one on each device, not having to jump around. So, oh, great. Why did it kick me out right now? I was in there before today. All right. Good. So when you go into the canvas under test 3, so that's the last module, so to speak, even though it's not called a module, on the bottom, all the way here, we have the test 3 review. And there you have the test 3 review, which is that physiology document, and then the term list again. And for the anatomy terms, like in the second test, if you felt the videos were helpful for you, then they are like here. That's pretty much covering all the terms you're going to do on the test. And that's that. So I have it then printed out right here. When you press on that, that's what you get. So today what we're going to do is we're going to go through the heart, through blood vessels, and through blood, and start on that process. So I'm going to basically go through all the different points on that document, and I want you, and I look at it, and I want to make sure you understand those points for sure. The rest of the stuff is also covered in the quizzes and in some of the labeling stuff. But for the physiology part of the test, those particular points are the ones that will be testable. And so it's also good to make flashcards out of those if you're a flashcard person. That way I deliver you the whole thing. You don't even have to worry about it. So can you guys see my heart?
Yes.
Yep.
Good. So the first bullet there is an ECG or an EKG tracing. And that measures the electronic, that measures the electrical impulses that are spread through the heart's muscle. And so that's very important for diagnostics and help us understand, certain problems in the myocardium, in the heart muscle. And so we have this EKG tracing, and ECG stands for electrocardiogram. And sometimes you see the cardio is a K, so it's sometimes you see EKG written. But you have a few remarkable things. You have a little uplift here on the line, on the graph. And so that's a discharge of electrical impulses, and that's known as the P wave. And the P wave is atrial depolarization. So let's just briefly go through the anatomy here. You've probably done that already. But when we look at the heart, we have blood coming from the vessels into an atrium. So those are venous vessels, what they bring in. Blue stands for how oxygenated the blood is. Blue does not stand for it being a vein or an artery. So this is why this looks reddish. And it's still a vein because it brings oxygenated blood from the lungs into the left atrium. So the atria are the 4 chambers, and then blood comes in here and dribbles down into the ventricle or gets pushed into the ventricle at the end, and that's what that P wave actually shows. Because an electrical impulse, an electrical discharge sort of goes together with the muscular contraction. And so you have to have an electrical impulse or a nerve impulse tell the muscles to contract, or the calcium to be released and then to attach to the myosin heads. I mean, the actin, and then that process of shortening can happen. And so that's where the ECG is helpful for many diagnostics. And so the P wave shows when this atria is contracting, or the impulse is telling those muscles to contract a little bit, and that pushes as much blood into the ventricle as possible. And then the next step on the EKG is when we have this big spike, the QRS complex, they call that. And that is when the whole ventricular heart muscle gets told to contract. So that's when that electrical impulse spreads through that. And when then that contracts, then that muscle shortens, and that internal content here gets squeezed out, and that gets then squeezed out through these ejecting vessels, which are the arteries, the pulmonary arteries or the aorta. We're going to look at some valves. We have a couple of valves here and between the atria and the ventricle, and then we have a couple of valves here between the ventricle and the ejection chambers. You can't really see these behind here, but there's the same set as here is underneath here. And so these valves are interesting because they're like flaps coming down, and they're attached to strings. And when that heart contracts and shortens, they lift up and they sort of close the door between the atria and the ventricle. And so they're called atrioventricular valve. And they, when the ventricle contracts, they assure that the blood doesn't go right back to the atria, because we don't want it to go there, but the blood goes and gets ejected through the ejection chambers, and the blood has to push open the other set of valves. They're opening upward. They're known as semilunar valves. That's the way they look. They open upward, and then the blood gets ejected, and when the ventricle, after a contraction, relaxes again, the blood that's still in these chambers here doesn't fall back into the ventricle, but then gets caught and lands on these semilunar valves. And so you don't have any blood coming back down. And so that's those. But that tells us about these waves when we look at the contractions. And I just also covered the valves because we have a question coming up after that. And so, so that covers the P wave for the atrial contraction, squeezing all the blood going into the ventricle. And then the QRS complex tells us when the ventricles contract. And then we have one more wave, and that's the T wave. And so when a nerve discharges, and we haven't touched the nerves yet, but when a nerve discharges, it's known as depolarization, because it has to do with ions moving around, and they carry around polarity. And so that's where polarization comes from there. And so a discharge is known as a depolarization. And once that's happening, you have chemicals that are scrambled, and they have to reorganize themselves and move across cell membranes again to re-establish this situation where we can have another discharge. And that's known as repolarization. And we'll talk about the specifics of that in the nerve chapter. But the T wave is sort of that recharging. If you call this a discharge of the ventricles, we can call this a recharging of that electrical capacity that the nerve can fire again. And you might ask yourself, why isn't there a repolarization for the ventricular, the atrial contraction? And there is, but it's hidden behind this big QRS complex. Whew. You got all of that?
Sure do.
Yep.
Good job. Well, then let's go right at it. See, this is actually a cool graph. This shows you everything, what's happening where. So this is kind of a consolidation thing, the cardiac cycle. So that's kind of cool. So what we have next on that review sheet is the systole and diastole. And that systole refers to contraction of the heart muscle, and diastole to relaxation of the heart muscle. And so you could see that number when you do blood pressure. You look at the higher blood pressure, that's the systolic blood pressure, and then you look at the lower number of the blood pressure. It's two numbers, right? And then what the heck? Sorry about that. Dinging. And then the lower number is the diastolic blood pressure. And so
I have a question.
Yes.
So when you talk about heart muscle, the heart muscle is just referring to the different groups of muscles that make up the heart?
No, the heart muscle is the myocardium. So that's the red part of the heart, is the heart muscle. And so it's one type of muscle. And so see that red part here, that's the cardiac muscle. But all of that is the cardiac muscle. And here you see a cross section of it. And the cardiac muscle is one group of muscle. It's very specific in the fact that it can contract as a unit. It doesn't, not each heart muscle has to be told by a brain nerve fiber that it needs to contract now. The muscle actually contracts as a whole unit. It has these, let me show you a picture that has these these intercalated discs, these gap junctions that let ions pass through, and then the nerve impulse can pass through the different muscle fibers and discharge through all of them at the same time. And so that's the difference with the cardiac muscle to the skeletal muscle, which is the stuff that we move consciously, like your leg muscles, your hand muscles, and all of that. And then the third type is the smooth muscle, and that's the stuff you find in tubes, like in the blood vessels. You have smooth muscle, and also in the gut, for example. So you have here, when you look at the chapter on vessels, you see here you have a tunica media, they call it, in a vessel, and that's smooth muscle. And that functions completely different. That's like a very slow contracting, more continuous, contracting stuff. You have that in the gut a lot as well.
So then the cardiac muscle, so there's different functions that are happening within the heart at the same time. Is that because what I'm understanding and what I know about what I understand about the heart is that it is made up of one kind of muscle, but there's multi-functions happening at the same time. So with
you mean like the electrical discharge and then the muscle contraction, that kind of differences?
Yeah, or like within the vessels, you know what I mean? Like I have a heart defect, and so I have an extra SA node, which makes my heart work against itself. So part of it's doing one thing and the other part of it is doing another. So it's the same muscle. So I'm still trying to understand that. So I was just asking a quick to see, I just wanted to know, like it's the same muscle, but it's like not doing what it's supposed to do.
Well, so if you have an extra discharge, you might get instead of a discharge, not a beat, but the discharge, at one discharge, you get the double, and the heart muscle probably gets somewhat confused a little bit with that.
That's okay. Okay.
So the electrical impulse talks to the heart muscle, and then that's sort of the stimulation that the heart muscle can contract. Okay. And then when the heart muscle contracts, what that does, it makes this chamber smaller, because it shortens and it contracts, and that pushes the blood forward in the one direction, from the atria to the ventricle, and then from the ventricle to the ejection chambers. And so your end result, what you want is the blood moving around the body, right? That's the main purpose of that. But to accomplish that, you have the electricity needs to talk to the muscle, and then a muscle can do its job, which is contracting the chambers.
Okay. So fascinating. What a piece of machinery.
It's extremely fascinating. And see, this is where you have it. You have the SA node here.
Yeah,
atrial node by the atrium. And so, and then you have the AV node here. Okay. And so probably you get an extra discharge or something like that. And maybe it doesn't always synchronize. maybe, you know, most of the time. But yeah, it's very interesting.
Wow. Okay.
And then the brain, and so do you have this internal heart happening, and then the brain stem controls that. So the brain, or I should say, influences that. So the vagus nerve and the sympathetic nerve, they influence what's happening. So one calms it down more. That's the vagus nerve, the parasympathetic, the rest and digest part of the nervous system. But of course, what we often feel much more is the sympathetic. That's when we get agitated and things get, you know, like I had one of the phone calls that I was anxious about this morning, and it's like I just feel it pumping heavier, and you know, that's the sympathetic cardiac nerve, and so they influence what's happening with the heart. So you have this intrinsic muscle system, intrinsic nervous system that does that discharge periodically, like 60, 70 times a minute in a normal situation, or, you know, generally speaking. And then you have the brain that can influence that underlying rhythm.
So one of the things about the vagus nerve that I learned was so sometimes my heart will just beat for like 240, like it'll just be at 240. Like I'll be resting, not doing anything, and my heart is beating in my chest like it's going to jump out. And the doctor that first figured out what was wrong with me said, bear down. He said, bear down, and it stops my heart. And my heart goes back to normal because he gets the vagus nerve. He like activates it. And that's the way that I use to get my heart from like jumping out of my chest, is I act like I'm going to take a dump.
Wow.
Stops it from like beating like, 240, beats per minute, which is crazy.
Yeah, that's way crazy. I mean, should be below 100.
Yeah, I mean
160, 70 is sort of the norm.
That's my baseline.
60 to 100 is the norm.
Yeah.
And above 100, we call it tachycardia.
Right. So I'm super ventricular tachycardic is what my condition is.
Right.
Okay.
But that's very interesting. So that's stimulating the vagus nerve to slow down everything that's going on inside.
It works.
Yeah. And that's amazing. That's amazing.
It works.
That's really amazing. Wow. Yeah, no, thank you for sharing that. I mean, that's very, it's, I'm sorry that it's happening to you.
No, it's fine. It's fine. It's a fascinating thing, but that's the direct connection for me that I just.
Absolutely. And then what also happens when the heart contracts, we have these sounds happening. So you hear the heart sound, and the heart sound is not blood going through or anything like that. It's when the valves close, that little clap thing, the lub dub, is when valves close. And so the lub is when the AV valves close, and the dub, which is the second sound, is when the semilunar valves close. And so I need you just to know that. It makes sense, when the heart, the ventricles start to contract, the AV valves could get closed up, and that's the lub sound. And then when the blood is ejected and falls back onto the semilunar valves, when it stops actually injecting, then we get the dub sound, and that's the second one. So that makes sense in that respect. And again, all here, it shows all of that stuff. So it's kind of cool how everything is mapped out a little bit. And then the last one is the cardiac output on this slide, on this part of the show. And cardiac output is how much can the blood pump in 1 minute. And it's a measurement of a multiplication between a heart rate. How many times does the heart pump per minute? We were just talking about that. And then a stroke volume, which is how much blood gets pumped each time it pumps. And at a resting, it's about 70 beats per minute. And the stroke volume is about 70 milliliter per beat. And that can double to about 140, and that can go up to about 180 if we need it. And so we actually, if we look at that, we pump about 5 liters of blood each minute as a volume, but we can go 5 times as much if we have to, run away from a lion or something really crazy like that. Of course, we can only do that briefly, because otherwise it's, it gets exhausted. So then we have some things on the blood vessels. So I alluded already before that the vessels that lead away from the heart are known as veins, and the vessels that lead to the heart are known as arteries. So here we have that right here. And that's because when you think about hydrostatic pressure wise, if you squeeze that hard and the blood goes out in this vessel, there's a lot of force. It's like a water hose kind of force. And that's, you need that wall to be a little bit thicker and a little more withstanding, have more collagen fibers in there, and make sure that it's strong. When you look at the vein, you have the same amount of arrangement. You have all these different layers, but it can be much boggier. It doesn't have to withstand that hydrostatic pressure. It's more like a vessel guiding the blood back to the heart. But it has different features. It, for example, has these valves that make sure the blood gets pumped, goes towards the heart. It doesn't get pumped. It moves towards the heart and not against, down towards gravity, away from the heart. And so you have these valves, for example. So you have different features, but they are different because they have different functions. And so has nothing to do with oxygen or no oxygen. It's just, it happens that most arteries are more oxygenated blood and most veins are deoxygenated blood, but not all of them. Well, the one not is that one we talked about briefly. It's the pulmonary arteries, which carry non-oxygenated blood to the lungs to get oxygenated, but that's an ejection vessel. And then the pulmonary veins is when the blood comes back from the lungs, and it's a vein, but it's oxygenated blood. And so that's the main difference where this doesn't match up. And then we have capillaries, and the capillaries are exchange vessels. So that's where we have leaky vessels. They are very thin. They don't have muscle layers around or anything. They just have an internal layer, and a lot of substances leak out into the tissues. And so stuff that the tissue needs leaks out, and waste products can come back in and be brought through the bloodstream. Then you can think of the bloodstream being like a highway system that can carry things around in the body and dump it in the liver or go through the kidneys and filter it out that way, the waste products, or through the lungs, breathing out the carbon dioxide, for example. And what's interesting about the capillaries, we have this ability to get blood perfusing that tissue, or we can shut it off and we just have a thoroughfare channel and not perfuse all the capillaries and all the tissue therefore. So it's a very intricate system of how we can control what goes where and the tissue being fed that needs to be fed. So it's really cool that way. And the other thing then becomes also a question mark when we think about, we have the heart that pumps blood through the body, and then in the capillaries, everything kind of seeps in and out of the tissues. And then on the vein side, who the heck is going to pump the blood from the vein back to the heart? And so we have a bit of a problem with that. In the vessels, we have the veins go through muscles. So when we contract muscles, that pushes the blood upward. That's kind of why we don't want to just be laying around couch potatoing that much. So skeletal muscles are very important. I already talked about, we have valves that help the blood not fall back down. And then also when we inhale, the pressure in the chest drops slightly, causing veins near the heart to expand, which then fills them up and brings the blood, sort of suctions the blood upward. That's kind of an interesting one. That's sort of the last mechanism, but they're not as strong as a heart. And so what we also have is we have lymphatic vessels, and they digitize these capillary networks, and they open only inward. So it's not a whole system like the cardiovascular system. Blood vessels go from the heart to the tissues back to the heart. This just goes from the tissue to the heart, and it picks up excessive fluid, and it has these inward channels and lets the lymph at that point, which is that fluid that gets pumped out of the blood. So it's cardiovascular stuff, it's heart, it's blood stuff, and it gets pumped back in. It just doesn't have the red blood cells in them, and it gets pumped back into the system near the heart, where it goes back in. We also know the lymphatic vessels because we got the lymph nodes, and so that's that's an immune system function, immune function organ, the lymph nodes, and so a lot of stuff that comes in is in the bloodstream, gets pumped out. We can sort of monitor what's in the tissues, but the negative effect of that is it also can spread cancer. So that's why lymph nodes become important. So when you have a person that had cancer and has lymph nodes removed, you can see how their legs or their arms swell up because this system doesn't work effectively anymore, or it's not as effective at that point. And so that's sort of where you can see that happening in real life. And then the last point there, lymphatic arterial pressure. Where is my pressure? Oh, a little brief discussion about all these different names of these vessels. Isn't that a cool picture? I think that was a really cool picture. You can see how the tissues are perfused. So the everything is taken away except for the arteries. But we have, when we look at these names, a lot of the names of the arteries, I put them here. We don't really test on them. Hepatic is liver stuff, and then we have renal is kidney, and so a lot of the names of the veins and the arteries are named according to the organ, or very heavily according in the extremities, particularly according to where they run. Like it goes under the axilla, so it's called the axillary artery, or under the clavicle, it's known as the subclavian artery. And then brachial, we know brachial, we've learned a lot about brachial, all these muscles. And radial artery, the radius by the thumb, remember, and the ulnar artery by the pinky. And then here we got a femoral artery, we got a tibial, and then and we don't have a fibular, they're a little behind, they still call it peroneal. The fibularis muscle used to be known as the peroneus muscle. And then maybe popliteal is a name for that's, you learned that in the first chapter with the body regions. And so, so that's the arteries, and the veins are very similar, except the veins go superficial, and then they have veins that go deep. And so we have a saphenous vein as well, and that's a superficial vein. And then we have a cubital vein. No,
I have a question.
yes, a basilic vein and cephalic vein. Mm-hmm.
So what's the distal artery of the foot? What was that one called again? I missed that one. What?
Dorsalis pedis.
The dorsalis pedis, the one on the inside of the ankle. Okay. Thank you.
Mm-hmm. Yeah, and I just put these as references because you can go endlessly in studying detail. But I think it's, you know, it's so similar to the bones very often that I didn't worry about testing on it. But the brain is interesting because the brain has a cerebral artery, and then it has a vertebral artery also. So the brain gets fed to the back of the brain, and then the front, the middle part of the brain, and then it has connections that actually circulate. They call it the circle of Willis. And so everything sort of has access points from two angles in case something happens. And so that's kind of cool, I thought. But then on the blood pressure, what I want you to know is that blood pressure is the pressure against which the left ventricle is the one that pumps the blood to the body, through the whole body, which the left ventricle must eject blood. And then, so that's what we measure when we measure blood pressure. How strong has that left ventricle has to eject blood that it goes through the whole body. And that measures blood pressure. And that measures how easy is it for the heart to pump blood through the system. Is it hard because the vessels are constricting? Is it hard because they have plaques in them? They're smaller. Or is it hard because there's more volume? Like, you know, if you have fluid retention, because maybe we do high sodium or something, we kind of feel the puffy a little bit. That can do something. And then yes.
What is there a reason why it's the left ventricle only?
Yes, because the right ventricle pumps the blood from the body to the lungs.
Oh, gotcha. Okay.
And then the left one goes to the whole body. And when we looked at the this picture, you see this is the left ventricle, the thickness of the wall, and this is the right ventricle. So the right ventricle doesn't really play that part in it because it doesn't have to be that strong. The lungs are right next to the heart.
Mhm. Okay,
Good. Very good question.
thank you.
And so then this pressure, where was my pressure wave? There it is. This pressure wave can be felt on superficial arteries, like in the wrist, you can feel it. You can feel it by the brachial on the middle of the arm, the carotid in the neck, or the femoral on the inner thigh. So there's multiple different places where we can feel that pulse and that pressure wave. That's that pulse. Good. And then that's the place where we can go to the blood. That's the biggest chapter here. So when we look at blood, blood is a connective tissue. It's fluid, it's not bone, but it's still connective tissue. So we still have a cellular component, and we have extracellular matrix, some components there. And the cellular components are red blood cells, white blood cells, and platelets. So we have all of them. So when we look at blood, we have the oxygen carrying cells, that's the erythrocytes. And then we have the immune cells, that's the leukocytes and the whites. And then we have the ones that cause coagulation in case when a blood vessel gets a cut and you're bleeding, we got to figure out how to not make you bleed out. So we got to somehow figure out how to heal that cut up. And that's kind of a complicated process because you have fiber. So when you look at the extracellular matrix down here, you have blood plasma, that's sort of the watery stuff, the yellowy stuff. And then you have soluble fiber in there. And soluble fiber means it's like, Metamucil or something. You know, you can dilute it in liquid and it's still fiber. And that's called fibrinogen. And remember, a GEN from the chemistry chapter, that means it's a precursor to something. It's like Genesis. And so that soluble fiber, when the blood vessel gets cut and we bleed out, needs to be put into an insoluble fiber, one that precipitates out in liquid. But we only want it right at the place of the cut. And we only want it for a very limited time, because if that insoluble fiber, which then is called fibrin, you take the GEN away, if that goes to the blood, then we get blood clots, and we don't want that because things get clumped together. They're going to block small little vessels, and that's a stroke, and we can't handle that. And so that's where the platelets come in, the thrombocytes, and also where the fiber comes in, the fibrinogen. So those are the components of blood. So we have that covered. And when we look at, then we go to the red blood cells. Here I put about, well, here you see the components of the whole thing. But this is about how much blood we have in the system. So we get a visual on it. But when we look at the functions of blood, we have, of course, oxygen and carbon dioxide transport. That's a big function. Then we also have a defense and immune system. So the white blood cells can defend against pathogens. So that's also pretty important. Oh, they're all important. And then we have transportation of other things like nutrients, waste products, hormones, etc. And another thing that's really cool is the heat distribution. So when it gets really hot, we can bring the inside blood that's really hot and boiling us up, we can bring it to the surface, and then it radiates off into the universe. And that brings heat away from the body, and that's kind of cool. And then again, we have that blood coagulation that we just talked about, the blood clotting, and that's sort of where you can see those fibers and the red blood cells and the platelets get caught in it, and then that patches up that hole that was created by some mysterious reason. Good. So that gets me then right from there into the white blood cells, the leukocytes. And we have multiples of them, and five different varieties. And we want to know a little bit about each different variety. And so the first one is the neutrophil. And let's go to the neutrophil. I'm not really There we go. That should be it. The neutrophil is the first one. And that is the one that shows up when there is a problem. So when you cut yourself, and then that is the one that shows up there and starts eating all the bad stuff, the bad material. And sometimes it really, lysosomes, have the enzymes in it that release, that can kill that foreign substance, but sometimes they perish themselves. And when they perish themselves, you have pus. So if you have pus, there was neutrophils, and that they took good care of you and they died for you. So if you feel depressed, then there's a reason to feel less depressed. Sometimes I have to tell myself that, but that's kind of cool, I think. So the neutrophils are ingesting type cells, phagocytosing cells that clean up the dirt that gets put into the body when we cut ourselves or so. And then the basophils are the next one. They release histamine, and that plays a role in a hypersensitivity reaction. So I want you to know that. Basophil hypersensitivity. Eosinophil are limiting allergic reactions and also fight parasites, parasitic worms. I know, the fact that we actually have parasitic worms in us. Oof. So that's the eosinophil. So I just need you to know a couple of these functions. That way you can differentiate those cells. And of course, for the lab part of we have the terminology where we have to identify them. But that's what you have in a homework. You worked on that already. I saw them. That's a pretty easy one, that one. And then we have the lymphocytes. And the lymphocytes are our B cells and our T cells. And they're part of this, what we call the specific immunity. And so we're going to talk about the immune system, but we have a non-specific immunity. So when you cut yourself, then you have an inflammatory process happening. You get red and you bleed a little bit, and that's non-specific immunity. And so that's sort of the first line. And then depending on what, can you mute somebody, please? Mute somebody, thank you. And then when bad stuff comes into the system and the body reacts to it, what the body reacts to is antigens. And we understand with the corona now, thank God, the teaching's already been done for me through the corona, because we talk a lot about the spike protein antigens. Can you mute? That then the system, our system, has to react to, because that's a ill-making, a sick-making component. And so our system then learns how to read that antigen and create antibodies. Can you mute? Lexi?
Here.
Mute. Thank you. Super. All right, there we go. Where was I? Then those the cells, those lymphocytes, can create either antibodies that are released from them, and then they neutralize the ill-making pathogens, the antigen part of the pathogens, but it takes the whole pathogen out. The antigen is just the reactive component of it. That's why these spike proteins are so important, because they are the ones that stimulate our immune system. And so the B cells are the ones that do that, and then the T cells can actually attack these specific cells as they have learned over time what an antigen looks like, and what, and then that one, they can attack that one because they specifically understand how that looks, how that shape is. So that's pretty neat about the B cells and the T cells, and it's called specific immunity because they specifically attack specific antigens. So they're very specific, and they have to learn that specificity. So when we get exposed to an antigen the first time, like in the old days, you got the neighbor has the chicken pox and the kids go all over to get the chicken pox. So that way, then you get exposed to it in an age group that doesn't make you very sick. And then your immune cells, your lymphocytes, learn how the chickenpox virus looks. And when you get exposed to it again, no problem. They are ready to go, and they have learned how that antigen looks, and they're already prepared to release a lot of antibodies and to attack it. So the B cells will release a lot of antibodies, and the T cells attack it. And that's why vaccination, that's the whole concept of vaccinations, is to prime that specific immune system, so the body doesn't have that lag period it takes for itself to be reacting to it, and it doesn't get that sickness going on. So that's the logic behind vaccinations, and I think, personally, I think in general, with that concept, it's probably one of the best inventions of modern medicine, because we're taking our own body function and we help it along the way. Of course, the problems is, I think the medical community sometimes has an issue communicating or having patients with questions that common concerns about vaccinations. And I do believe they vaccinate the heck out of the kids these days, and that can be, I think, a little overwhelming for the system. So what I did with my kids is I just asked the doctor, how long can I wait with one or two of them, and try to have my child be as large as possible, and have as much volume and as tall as possible at the time they get the vaccination, so there's more volume that the body can deal with it. But anyway, I mean, because in California you kind of have to if you want to send the kids to school. But that's the logic behind it, and that's also why they're urging us to try to get the vaccine if we can for COVID, because the more people that can react to COVID that way and not get sick, so that the virus doesn't have the ability to replicate the whole time, then it doesn't mutate as much, because you need replication to have mutation. Anyway, but that's the lymphocyte system, so that's pretty cool, and we'll talk a little more about that when we get to the immune system. And then we have monocytes, and again, if you have questions, just ask, don't hesitate. If we have monocytes, that's the last of the brands that we look at, and they are cool because they leave the blood system and go to tissues and become resident macrophages. And macrophages or phages are eaters of big things. And so they are actually phagocytosing, destroying bacteria and parasites and all kind of stuff. And like in the lungs, they call them the dust cells. In the liver, they call them the Kupfer cells. In the skin, they call them the Langerhans cells. So we're going to have all these names come up, but I like the lungs the best because dust cells reminds me of a vacuum cleaner. And so that's kind of what they are. And so they take up, yes.
I'm sorry to interrupt.
No, it's good.
But as you're describing it, it helps me understand and kind of categorize each group of terms.
Mm-hmm.
So
absolutely.
Sites, when you say, I understand mono and then sites, and we have, you were saying about the other sites. What does that mean? The sites part of that?
Oh, that means cell. Cytology. Sites is cell.
Okay, gotcha. Okay.
So SYTES is cell.
Okay, and then the five.
Yeah, and I'm not sure why they call it monocyte. Maybe somebody have read up on that, but I think I did a long time ago, but I forgot about it. Yeah, keep going. Sorry.
Oh no, same thing. I don't know if there's like a list. I'm sure I can Google it. A list of all of the different terms, like the monocytes, and then you have the macrophages you just mentioned, and then the phagocytos, right? And
yes, so, you have a lot of that terminology, and that's probably a good one to write out and learn, because they're very repetitive.
Right.
Phage means eating or ingesting. Macro means big stuff. It's not micro, it's macro.
Okay.
Like macronutrients.
Okay.
Mm-hmm. And then what else was there? We do antibodies, antigens. We have talked about that for quite some time in our language now. Neutrophils, I'm not so sure what the neutral stands for. That's a good question there. And I forgot Phil, actually, too. But the funny thing with the neutrophils is when you look at the lab component, it's sort of the background of the cell is similar to the nucleus. And so I feel like it's neutral.
Yes. Okay.
Right. That's how I remember. Or the basal or eosinophil. For some reason, eosino is like reddish, is like neon, you know,
Right.
bright, and baso is like, you know, basic. It's not acidic, so
Mhm.
it's bluish, and so, you know, the staining kind of helps me around with those from a
Gotcha.
perspective of recognition.
It'll help me remember. when-
No, we need, that's very important to have those. You know, the brain does not work in this abstract. It works in visuals and pictures and mnemonic
Right.
and stuff like that. Cool, cool, cool. So those are the white blood cells in our cells, and then we have this brief discussion about platelets, which we already kind of did talk about. Well, actually, in the platelet discussion, what I'm really concerned about is what they are used for, and that's the blood clotting. And so when we look at blood clotting, we kind of need to look a little bit at step by step what's happening. So the first thing when it happens, when you cut a wall, when you bleed, when you cut yourself, we want to slow down the bleeding by vasoconstricting the vessels that go to that area. We call that hemostasis. And then the second thing that we want to do is the thrombocytes are coming out. These platelets, thrombocytes are platelets, and they get deposited along the vessel wall and stick to one another. So they create a thing, we call a platelet plug through that process. And then after that, we have the blood coagulation coming next. And this is a very important step, because we have blood cells get brought, clumped together, and get also brought into this meshwork where we first started with platelets. But in order to do that, we need to have insoluble fiber, and because the fiber can stick it all together. And so that's when we get fibrinogen going into fibrin. And so the fibrinogen is the soluble fiber that doesn't do any damage if it's not needed, and the fibrin is the insoluble fiber that gets the clumping going on. And so we need to be very careful how we do that process, that we don't create a floating fragment, which would be an embolus, which could give us a stroke. And so that's then the fourth step that we have. And so we have those four steps that we can kind of differentiate one another from. And that's one of the points that I want you to understand, to differentiate those four steps. We don't need to go into, there's a whole bunch of detail, thrombin to this and that. I'm not worried about that. I just need you to understand that last step between the insoluble fiber and the soluble fiber. I think that's enough data to get a decent understanding of that. And then, you know, for you guys who go to Bio 2 or so, you get more into detail on the process. But believe me, at the end of 5 years or whatever, you will be basically remembering this part again. And I think that's very good enough. All right, and then we have a couple of points on lipoproteins. I want to make sure you understand that the LDL is the lipoprotein that carries lipids from the liver to the body tissues. We know that as bad cholesterol, and the HDL is the angel because it carries the lipids back from the body back to the liver. And we call that good cholesterol because it helps take away the lipids that we don't necessarily want to have excessively, or the ones we don't want. It brings it back to the liver, and the liver can process it. So that's good. We need to know that the hemoglobin in the red blood cell is what carries the oxygen molecule. And then we get to the good old blood groups. And I'm interested in you understanding the ABO system. And the way that I look at, you know, we have a different blood type. And what happens, the reason we need a blood type is that we have on our own cells, we have antigenic markers. Now, our own body can be cool with that because it's been living with that for eternity. But somebody else might not be cool with that because their blood cells might not have those specific markers. Antigens do not have to be negative. They're just generally speaking bad because they're on pathogens and we think of them as such. They're also self-markers, but if you give, for example, a person who is a person group A blood, that is actually a group B person, they have a reaction to that because their markers on their red blood cells do not work together with the markers in the recipient's blood. And so what we do, we have two different markers that we in the ABO system that we really worry about. The markers are A and B. And so the groups are named by the marker. So the antigen is the marker. So in group A, we have antigen A present. And what that body does, it has antibodies to the B antigen, so the opposite. So it has antibodies to the opposite blood type. So that's, and then you have group B, and that's the B antigen, and that blood has antibodies to the A antigen. So the problem is, if you give a B person A blood, you're going to have an antibody against that A, and you're going to interact with that, and you're going to clump that blood together, and that's not what we want. The agglutination is a problem. And so we want to make sure we give a group A blood and a group B blood. And then we have a couple more compositions here. We got one blood type that's A and B. And the cool thing is that group has no antibodies to either A or B. So that group is the universal recipient and can get blood from anyone. So that's kind of nice. And then we have one blood cell that has no of this A or B markers on it. There is other markers too. They just don't interact. It's just the A and B interact heavily. And so the group O has no, O as in zero, kind of, no antigens. The problem is they have both types of antibodies. And so this blood can be given to anyone, which is very cool for anyone, except the O people can only receive O blood because they have both markers, and so they interact to everybody else. You got that?
Got it.
Good. I was concise. That's not always an easy one to figure out. So I'm glad you got it. So if you don't have a question, what I have is a couple more questions. I want you to take out a sheet of paper and I want to go through a couple of heart terms with you to finish up. And then we're done in a couple minutes. We're only on the heart. Where is my heart? Picture. There you go. Ha! There's my heart. Can you see my heart?
Yes.
Oh, there it is. So if you take out your term list, you got your term list too? We're going to start working on the first column there under the heart. And what I want you to tell me or write down are a few terms here. I'm going to start slow. So first term I want you to tell me is, what is this vessel called that brings blood into the right ventricles from the body? There's one from below, which is behind this flap, and one from above. Just give me the general term of them. The vessels that bring the blood to the right atrium. What is that vessel? What are those vessels called? That's number one. And then number two, when we look at the left ventricle, the blood gets ejected up into this big round artery. What is that called? Actually, what is that part of that artery even called? That rounding. Now I've got to be quiet, otherwise I give it away. And then number three, I want you to give me the name, the generic name from the valves. Both of them have the same generic name, the valves that are between the atria and the ventricle that close up that passageway when the heart contracts. And number four, last one for this today, we make it very gentle, is I want you to tell me, what is the wall called between the two ventricles? So there's a wall that's between a muscular wall that's between these two sides of the heart. What is that called between the two ventricles? Good. And so let's solve those little questions. So I want to have as many voices as possible so I don't feel all lonely over here. What is number 1?
Aortic arch.
Vena cava.
Vena cava.
Vena cava. Good. Number 2 is what you said.
Yeah, I got
Aortic
it.
arch.
Aortic arch.
Aortic arch. Perfect. So the vena cava, we have one on the top, one on the bottom, brings the blood to the right atria. And then we go through to the ventricle. What valve do we pass? Number 3.
Intraventricular.
Ventricular valve.
Very good. Atrioventricular valve. Excellent. And then number 4, what is this wall called?
Intraventricular
Ventricular
septum.
septum.
Excellent. Interventricular septum. Good. See, the term says it itself. Inter means in between. Ventricular means ventricles. And so as long as, if you go through these terms, I think it's very helpful and important probably too, to slow down and be slow about it and look at the word. What could it mean? And then again, as I forgot who was asking and talking to me about it, when you look at these terms, phagocytosis and all that stuff, accumulate the knowledge like it's a vocabulary of a foreign language. All right.
I'm sorry.
Anybody?
What did you say number 3 was? You said not number 3. Yes, it was number 3. The
Atrioventricular valve.
atrial ventricular valves. Okay. Thank you.
Good, good. All right. How is it going over there? You guys have some questions for me?
I do have a question about the blood pressure.
Mm-hmm.
When you take your blood pressure and the high number, it's about 120, which it says is normal, but the bottom number, what is it, the diastole? It's lower than what it should be. What does that mean? For example, 130 and 60 instead of 120 and 80.
Yeah, we're more concerned about being too high. If it's 60, that doesn't. I don't know the exact numbers, but that seems to be still high enough to not be a problem.
Okay.
So yeah, no, I don't. We usually start worrying about it if the lower number goes above 85.
Oh, okay. So it's better to be lower than to be higher, I guess.
Yeah, I mean, if you're really low and you don't have, you got nothing over nothing, then the heart ain't pumping. So that will be a problem. But generally speaking, we want to have the blood pressure not go too high. And that brings me actually to the last point that I almost forgot to mention, and that's the sodium discussion, the blood pressure discussion. I want as an exercise, read through that blood pressure handout that I have in a health kit. But as an exercise, I want you to look at some of the sodium intake you're doing, and measure it a little bit for 1 week. Do the best you can. Read some labels and see what's on there. If it's vegetables and stuff, vegetables is not the big sodium problem comes in from the processed foods. And so, you look some up, but do some tracking for a week and see where your sodium intake is, because blood pressure is something that our society has to grapple with, because we have a lot of things that cause higher blood pressure, and sodium is just one of those things. And I think if you get some awareness around that, that might be helpful.
All right. Okay.
All right.
Thank you.
Cool. Thank you. So let me take some rolls and then we can go. And then if anybody has something, just speak up. So I got Tanaya.
Here.
Anna just had. Sarah is here.
Here.
Vannery.
Here.
Kayla May.
Here.
Nita.
Here.
I got Koa.
Here.
I got Jordan.
Here.
I got Chamuna.
Here.
I got Brianna.
Here.
I got Berenice.
Here.
Berenice, you're here. I got Nelia.
Here.
I got Caitlin. And I got Lexi.
Here.
Good. Well, thank you so much for paying that much attention. And I hope have a good one.
Thank
Bye.
you.
Thank you.
Thank you so much. Bye.
Thank you.